أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم ولو نشاء لطمسنا على أعينهم فاستبقوا الصراط فأنى لهم يبصرون ولو نشاء لمسخناهم على مكانتهم فما استطاعوا مضيا ولا يرجعون وما نؤمر ننقص في الخلق فلا يعقلون وما علمنا شيئا ما ينبغي له إن هو إلا ذكر وقرآن مبين لينذر من كان حيا ويحق القول على الكافرين. Sadaqallahul Aliyyul Azim. Lecture number 9 for 11th class, and today a discussion how energy is produced. How energy is produced in our body. Very complicated mechanism. One chapter of respiration is allocated for these informations, but today I will give you some initial informations which are required to produce the energy in the form of ATP, and especially organelle, which is such important organelle in the cell. If we have energy, we are speaking, we are walking, exercising, and our food is being digested, the heart is pumping the blood to all parts of the body, in the lungs oxygenation taking place, the kidney nephrons are working, the blood is flowing with the speed, the blood is coming with the speed, all metabolic reactions taking part in the liver, and even the lymphatic system is on the right track, just because of that energy, and that energy just because of that organelle which I am going to discuss today. I think if I will say that in cellular life, this is the heart of the cell. One is the heart in your body. I am talking about cell. The cell as the heart, the mitochondria, because that is going to produce energy, because heart in your body produce the blood. Supply the blood to your body, and that blood carrying oxygen and produce energy. Similarly, this organelle is a specific organelle, and that organelle is a true organelle which is producing the ATP molecule. And ATP molecule formation is so complicated mechanism, because the students, you have to read almost 3 months in the senior, when you will become senior, when you will study at bachelor level, and you will think how the energy is produced. So I have to summarize now, and in that chapter I will tell you little detail, and two membranes I will discuss: outer mitochondrial membrane, the inner mitochondrial membrane. And outer mitochondrial membrane is not important. I am talking about the mitochondria. Outer mitochondrial membrane is not important. Inner mitochondrial membrane is acutely important, because that is folded. Just like small intestine was folded, just ileum was folded. What was objective to fold the intestine? The objective to fold the intestine to increase the surface area, because when surface area will increase, mostly the absorption will take place. We go to first slide today. This is my first slide today, and that will explain. Yes, I had already started from inner membrane of mitochondria. Inner membrane of the mitochondria, cristae. The name is the cristae, C-R-I-S-T-A-E. Spelling you will see, A is written, this is S, C-R-I-S-T-A-E. Cristae is backbone of the structure of the mitochondria. So backbone of the cell is the nucleus, but the heart of the cell is the mitochondria, and the backbone of the mitochondria is a cristae, which is inner membrane. Look here, this is membrane, outer flat membrane. I will show you figure after some time, and then the inner membrane is folded. Why the foldings are there? Why the folding? To increase the surface area. These are so organized functions, so organized that when you study, you develop interest. Because I did not tell you the proton movement, how the proton is moving into inner space, between outer and inner space, how this mechanism is created. Nature knows this only. We are unable to understand, but it is very interesting how this energy was being produced. Just like you are inventing a machine and you are just producing energy, how this machine will work. And energy is the main issue in all processes, in all inventions, and this human being is a such machine. That energy is produced in a super manner. I will say. Why the super manner? Look here, I am going to highlight ETC, electron transport chain. That is the electron transport chain. So ETC mean electron transport. I'm just watching. I just wanted to indicate now. This is ETC. Black indicator is telling that is the ETC. ETC means electron transport chain. Why the electron transport chain is required? Energy. Energy to produce. Just there are 7 kids are standing at one place. The one is catching the ball and throwing the ball to other, and the other is throwing the ball to next one. The next one is throwing the ball to next one, and this chain is created. This chain of energy creating mechanism, when I will brief you in the coming chapters, and you will see that how these kids like molecules moving, getting the energized electron, flowing the electron from the one place to another. It is moving from the one individual to other individual, to other individual, to the third individual, the third individual to the fourth individual, and they will produce the energy in such a format that energy in organized manner will be given to proton. Interested here? If you will just see the proton H+1, it will capture the energy, just like a rat and cat game is going on in your cell. Amazing that it will get the energy. Proton got the ball. It is moving. Where it is moving? To inner space, outer membrane space, between inner membrane and outer membrane. And what happens in between inner membrane and outer membrane? Inner membrane, I told you importance, reason surface area more energy will be produced. That why this is question, sir, surface area more energy will be produced. How you will prove by diagram? I will prove in the chapter of respiration. I think this chapter is included in first year. Just give me time. Yes, this chapter. Luckily, you have this chapter. You have this chapter in your book, and you can see that how the ETC will be able to produce energy. The proton got the energy now. The H+1 got the ball now. Where the H+1 is going now? It is going to space between outer membrane of mitochondria and the inner membrane of mitochondria. The space between outer membrane, inner membrane, H+1 gone. Who pumped the proton? Who send H+1 to this space? Proton pumps. You will study that chapter. The proton pump help get this energy H+1. Go. Nobody can catch you. This is a game. The H+1 is going to the space between outer and inner membrane. And then what happens? ATP synthase called. Where the ATP synthase situated? Please hold on. If you are not attentive, I can stop, because this is important information of today's session, the important conversation, the important sentence, the heading of today's session I am speaking now. What is the heading now? ATP synthase enzyme, which is written in your book as F1 particle. That is not F1 particle. That is just name F1 particles. Basically, they are ATP synthase enzyme. They can produce the ATP. How they will produce? They will call Mr. H+1, I want to get energy from you. Would you like to give me energy? Would you like to give me? H+1 says, no problem. I am hiding in the between outer and inner membrane space. You can come and just collect the energy. Amazing. So ATP synthase enzyme will get the energy from the H+1. H+1 will combine with NAD plus and NADH. Energy will be released. That energy will be used to form the ATP. ADP plus P will combine, and that chemical reaction will be facilitated by ATP synthase enzyme. What is the word which you will study in the heading of the mitochondria? F1 particle. We go to this slide once again. The backbone of mitochondria is a cristae. Spelling you will correct. I will speak the spelling CRISTAE. Cristae. ETC mean electron transport. This is the rat and cat game I am talking. ETC one is giving. How it will happen? I just show you diagram once again. I will try to explain little bit, because you know that is not our topic. General information about mitochondria, that ETC topic you have to study separately, and in 3-4 lectures I will brief in the coming chapters about the ETC. In short words, I told you that it's a chain of the molecules. They are throwing the energized electron. The question is from where the energized electron come. Everything will come in the sequence. We go to next slide, which I want to show today. That is the membrane of the vacuole. One important organelle. I think that is the organelle which is present in the cell, due to which you have the water storage, water tank in the cell. The water tank is required in each house. The water tank is required in your body. Water tank is required in your cell, and that is the vacuole. And vacuole membrane is the tonoplast. The vacuole membrane tonoplast, special membrane. It will allow some molecules to pass through it because that has the semi-permeable nature. It will not allow all molecules to pass through it. It will allow some molecules to pass through it. It will stop some molecules to pass through it. So tonoplast, now this slide you are watching, now I come back to a vacuole which is just like a stomach. You are eating. If you are eating, where your food is being digested, tell me. In the stomach. Where your food is being digested? In the stomach. When the cell is eating, where the food is being digested? In the food vacuole. The cell is small living organisms. Look at the units, millions, billions, trillions of the cells your body consists of. Each cell is just like a living thing. And each cell vacuole behave like a stomach. Reason, because that food vacuole will digest the food. Do you remember I was talking about the lysosomes? The lysosomes was having the digestive enzyme and Golgi vesicle with the digestive enzyme. Such a Golgi vesicle with the digestive enzyme, that is called the lysosome. And lysosome combine with the food vacuole to form the secondary lysosome. Why it was combining? But question is why it was combining? It was combining to produce, just want to highlight something, yes. To produce the digested food, to digest the food, we'll go back to food vacuole, contractile vacuole, the cell sap in the vacuole. This word is mostly used for the plants because you know nitrates are soluble in the water, and the water plus the nitrates, they will move up by the stem, by the xylem, and that will be collectively called as a cell sap. Cell sap is important because cell sap is providing the nitrates. I'm just talking about the type of the vacuoles. The one vacuole I told you, the food vacuole, that the food is being digested. It is not a stomach. Yeah, stomach is something else. That is the cavity in your body which is digesting the food in the protein. You will study the digestive system also in this book. Food vacuole is the stomach of the cell. The food, cell need energy. How the food will be digested in the cell? By the food vacuole. How the food vacuole will get the enzymes from the lysosomes. It is not digestive system, it is something else. I am talking about the unit, the cell. One cell in your body, food vacuole, consisting of enzymes. The enzymes are importing from the lysosomes. The lysosomes will send the enzymes from lysosomes to the vacuole. The vacuole will get the enzymes. When it will get the enzymes, what will happen? Protein changes to amino acid, the fats changes into fatty acid, glycerols, and the starch changes to glucose. And that digestion completed, it means your cell will get the energy by help of which organelle? Mitochondria. Mitochondria. So mitochondria need to react. Now question is, sir, vacuole producing energy? Yes, question from your side. Sir, vacuole, that you are discussing two major heroes. One is a vacuole, one is a mitochondria. Sir, I think vacuole is producing energy because food vacuole is digesting the food. And no, because food vacuole is just producing the glucose. How the glucose can be converted into energy without electron transport chain? That is the process you have to study. Electron transport chain take place in mitochondria, and electron transport chain take place in inner membrane of the mitochondria, mean cristae, and then the proton will move into membrane space where this rat and cat game is going on in mitochondria, and that is producing by ATP synthase, which I was referring F1 particles. Go to next slide, students. This is the important slide you are watching now. I should remove my picture from here, little bit yes side. I move at a side, and then indicator I require, I will little magnify. Yes, the most important structure in the mitochondria is the F1 particles, visible to you now clearly. The same thing I was talking about, that F1 particle, you will think that is ATP synthase enzyme. And ATP synthase enzyme I have to highlight. You want me to highlight? Okay, no issue. I will highlight here. This is the ATP synthase enzyme. Black indicator is telling you where it is used. Where it is used. ETC. The word is written ETC. ETC mean electron transport chain. Electron transport chain. What is electron transport chain? Once again the question coming. This is a full topic, and I think 3-4 lectures will be required, but I will tell you once again in a simple way. This is a movement of energized electron from the one molecule to another molecule to the another one. Question, sir, why the energized electron, where that was moving? I want your attention, please, because if you will miss this point, if you will get this point now, you will be very easy in that ETC when I will teach you in the coming chapters. Okay, now listen me carefully. The electrons are moving from the one membrane, from one molecule to another molecule. In which place of mitochondria this transfer of the energized electron is going on, Christe? I was giving importance, you noted at the start of my lecture, I was giving importance to which part of mitochondria? I was giving importance to which part of the mitochondria? I was giving importance to inner membrane of mitochondria. I was giving importance to F1 particles. I was giving importance to ATP synthase enzyme. In ATP synthase enzyme, which is present in the inner membrane of the mitochondria, that will do this ETC. Question, so why this ETC is becoming why this ETC is essential? It is producing energy, because energy will be released by this ATC. All procedure will tell you later on, but I will tell you, because it is moving from, it is a redox reaction, oxidation reduction, electrons removal, oxidation, electrons addition, reduction. This oxidation reduction reaction will release the energy. That energy will be given to the rat is proton run, and because it gets energy, goes to between inner membrane, outer membrane space, and then ATP synthase enzyme call, please give me energy, and ATP synthase gets H+1, and H+1 agreed to give him energy, and that energy was used to combine ADP plus P to form the ATP. Simplest way I explain how this game is going on. Go to next slide now. No, there is no slide now. There is the information which I have already given you, and you will see these information. I have to magnify the diagram now. Yes, this is the diagram of the mitochondria. Sorry, because I just want to magnify first of all. Magnification is there. Now you can see very easily. The magnification of the diagram is there. Now that is very easy. You can see now. I have to highlight now F1 particles. Okay, hold on. Yes, these are the inner membrane. Black color. Once again. Okay, see here. This is the inner membrane. Visible? I highlight once again. This is the inner membrane. Visible? Let me tell. Okay, this is the inner membrane, and this is the outer membrane, which is not important. Now I know what you want me to tell. You just wanted to know where is space where the proton run, because H+1 was running after getting the energy from the ETC. This is the space where the proton comes. Sorry, because I was unable to highlight. So I have to show you once again. This is the space. Yes, this is the space between inner membrane, outer membrane. So where the ETC taking place. Inner membrane, this is inner membrane. This is inner membrane. This is inner membrane. This is inner membrane, and ETC take place in this membrane. That's why it is folded. You are watching foldings. I am folding now. Yes, these foldings are just to increase the surface area. Now you got the idea why the surface area will be increased? To increase the ETC level. More electron transfer, more energy will be produced. You just wanted to know, sir, where the proton was gone. Proton gone there. So where is the ATP synthase enzyme? Yes, I told you, F1 particles or ATP synthase enzyme. F1 particles, where it is labeled, yes, this is the labeled. Where is the labeled? Now you can see, I just highlighting where is the F1 particles. Visible to you? Yes, this is the F1 particle. These are the main parts, because they are able to capture. Are you able to capture the money from your cousin, because cousin got the money in the pocket and you just want a party and just convincing your cousin you will be able to get or not? This is your technique. Similarly, this ATP synthase enzyme is able to convince H+, please give me energy, H+1 got captured, and he gave energy to ATP synthase enzyme, and ATP synthase enzyme combine where ATP, yes, this is the question, in the mitochondrial matrix. I am just going to highlight them in the mitochondrial matrix. Where is the mitochondrial matrix now? Yes, this is the mitochondrial matrix. You are visible, and now I think you got the idea of all this. One information I have to give you: porins. Anything which enters mitochondria, like nucleus, they have a protein called porins, and these porins protein allow some molecules to pass through it. Some molecules will not go. So this is about the now very simple information about the. Very simple information about the vacuole. Two vacuole, plant vacuole very large size and animal vacuole small size, and tonoplast is mentioned. Tonoplast, this is the membrane of the vacuole where the water will be stored, the food vacuole. Plant vacuole is large, animal vacuole is the small. Storage of the water, digestion of the food, and removal of, I did not tell you contractile vacuole, I think so, contractile vacuole. Contractile vacuole, contractile vacuole. I have to tell you about contractile. It is not written here. Yes, because I just want to show you from your data as well, because you will be able to highlight. This is the contractile vacuole. Okay, ji. Contractile vacuole is not present in all living things. It is present in amoeba. You know that, where is to remove excretory products. Sir, we have no contractile vacuole. Why nature has not given us contractile vacuole? Yes, because you have the kidneys. You have the kidney. If you are watching the second year lectures, I'm just briefing that the kidneys are the main excretory and osmoregulatory organs. So no need of contractile vacuoles in your cell. Thank you very much. This was lecture number 9. I hope you will revise and you will get useful information. Thank you. Allah.